This video provides an overview of the different steps in glycolysis. Biological organisms require energy to survive, and adenosine triphosphate, called ATP, is a form of energy that is used to drive various functions throughout the body, such as active transport, metabolic reactions, energy transport, energy storage, and muscle contractions. One way ATP is formed is through the process of glycolysis. Cells use glycolysis to transform sugars, like glucose, into biochemical energy in the form of ATP, NADH, and pyruvate molecules. These energy molecules may then be used to create even more energy for the cell by entering processes such as the citric acid cycle. Overall, the process of glycolysis is an anaerobic process that yields 2 ATP molecules per molecule of glucose that is input into the cycle. This process is considered to be anaerobic because it does not require an oxygen input to take place. In fact, the ATP production in glycolysis acts as the sole source of energy for some anaerobic organisms, such as certain bacteria and fermentation yeasts. The process of glycolysis takes place in the cytosol of the cell through a series of 10 enzymatic reactions to convert sugars into energy molecules. I liked this image because I thought it nicely illustrated the 2 energy consumption steps at step 1, right here, and step 2, or 3, right here, where you can see that a phosphate group was removed from an ATP molecule, leaving behind ADP. Let's take a look at an overview of the key steps of glycolysis, focusing on the input and output molecules for the entire process. The first 5 steps of glycolysis convert a 6-carbon sugar, called glucose, into 2 3-carbon sugars, called glyceraldehyde 3-phosphate. These 2 glyceraldehyde 3-phosphate molecules are generated in steps 4 and 5. However, in order to form these molecules, ATP is consumed in the first and third steps, causing this first half of glycolysis to be known as the energy consumption phase. Both of the chemical reactions that occur in energy consumption steps 1 and 3 are catalyzed by a kinase enzyme, and both reactions are irreversible. The next 5 steps of glycolysis produce energy and therefore make up the energy production phase of glycolysis. In this phase, 1 ATP is made for each of the 2 glyceraldehyde 3-phosphate molecules that were formed in the energy consumption phase. This means that a total of 4 ATPs are produced in glycolysis. However, 2 ATPs were used during the energy consumption phase for a net ATP production of 2 molecules. Major aspects to look at in this phase is that the ATPs are formed from the glyceraldehyde 3-phosphate molecules in steps 7 and 10, and both reactions take place via a kinase enzyme. However, it is important to note that step 7 is reversible, but step 10 is not. Another important fact is that NADH is formed in the 6th reaction that is catalyzed by an enzyme called dehydrogenase. NADH is an electron carrier that has the potential to produce more ATP energy molecules through another process called oxidative phosphorylation. Overall, the process of glycolysis results in a net production of 2 ATPs, 2 NADHs, and 2 pyruvate molecules. Here is a layout of each enzyme that catalyzes one of the 10 reactions in glycolysis in the order in which they occur. So here you can see the input molecule, glucose, and the following enzymes in the 10 different steps to get to the end product. Now that you've had an overview of glycolysis and know the important steps to take note of, let's delve deeper into the specifics of each of these 10 reactions. We start out with 1 glucose molecule that has 6 carbons on it. This 1 glucose molecule will be oxidized into 2 pyruvate molecules by the end of glycolysis. In the first step of glycolysis, A kinase enzyme catalyzes the phosphorylation of glucose at position 6 to form glucose 6-phosphate. The phosphate that is added to the glucose molecule comes from ATP, which therefore converts the ATP molecule to ADP. Remember, this is step 1 of the 2 energy consumption steps and is also an irreversible reaction. In step 2 of glycolysis, an isomerase-mediated reaction converts glucose 6-phosphate into fructose 6-phosphate by rearranging the covalent bonds in the molecule. Step 3 uses another kinase enzyme to catalyze a phosphorylation reaction. This step removes another phosphate group from ATP and gives it to fructose 6-phosphate to form fructose 1,6-bisphosphate. The ATP molecule used in this step is converted to ADP through the loss of one of its phosphate atoms. It is therefore the second irreversible energy consumption step that occurs in glycolysis. In the fourth step of glycolysis, a lyase reaction cleaves the 6-carbon fructose 1,6-bisphosphate into 2 3-carbon sugars, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. In the fifth step, dihydroxyacetone phosphate is rearranged by another isomerase to form a second glyceraldehyde 3-phosphate molecule. At this point in glycolysis marks the end of the energy consumption phase. By this stage in the process, the original glucose has now been metabolized into 2 glyceraldehyde 3-phosphates and 2 ATPs have been consumed in steps 1 and 3. Now, we'll delve into the energy production phase. In step 6 of glycolysis, both the glyceraldehyde 3-phosphate molecules are oxidized to 1,3-bisphosphoglycerate by a dehydrogenase enzyme. As highlighted in the process overview, this step also produces 1 NADH molecule for each of the oxidized glyceraldehyde 3-phosphate molecules for a total net production of 2 NADHs. These NADHs are later used to produce more ATP for the cell and act as the reduced coenzyme that is used to create more energy in the electron transport chain. In step 7 of glycolysis, another kinase enzyme catalyzes a phosphorylation reaction. The kinase transfers 1-phosphate from 1,3-bisphosphoglycerate to ADP to form ATP and 3-phosphoglycerate. Here, it is important to note that this step is reversible, even though ATP is formed. The 8th step of glycolysis involves a mutase-catalyzed reaction that moves the phosphate on the 3rd carbon of 3-phosphoglycerate to the 2nd carbon position to form 2-phosphoglycerate. In step 9, a lyase enzyme catalyzes a dehydration reaction, which removes water from 2-phosphoglycerate to form phosphoenolpyruvate. The final step of glycolysis, once again, uses another kinase-catalyzed reaction to remove the phosphate group from phosphoenolpyruvate. This phosphate group is then donated to ADP to form another ATP energy molecule in pyruvate. Here, I will highlight that this step, like reactions 1 and 3, is irreversible. Now that the entire process of glycolysis is complete, 2 pyruvate molecules, 4 ADPs, and 2 NADHs in total have been formed for each glucose molecule that entered the process. However, due to 2 energy consumption steps, the net production at the end of glycolysis is 2 pyruvate molecules, 2 ATP molecules, and 2 NADH molecules. The pyruvate and NADH molecules could now be used as inputs into other energy-producing pathways, such as aerobic respiration, to produce even more energy for the cell. It is also important to note that, in this video, glycolysis is being demonstrated as a closed process, but in cells, substrates produced by other reactions can enter glycolysis at different points. For example, when an animal breaks down glycogen, glucose 6-phosphate is produced and can then enter the glycolysis pathway at the second step. This could be beneficial because this means that one less ATP is used in the pathway because the first ATP consuming step in glycolysis is skipped. The other sugars can also enter the glycolysis pathway at different points and would each have different effect on the net number of ATPs that are produced by glycolysis based on where they enter in the pathway. So, now that we have delved into one of the major pathways that produces energy used in many biochemical pathways, such as metabolism in an organism, I hope you are able to understand the major inputs and outputs of glycolysis, the reactions that take place in the process, and why glycolysis is so important to sustain life. Other pathways that generate energy in eukaryotic organisms include the citric acid cycle, also known as Krebs cycle, and the electron transport chain, also known as oxidative phosphorylation pathway. Thank you for watching this video, and I hope you found it helpful.